Welcome you all. Today we are going to see about plasmids, it is particularly PBR 322. I am Dr. M LENA, Assistant Professor, Department of Zoology, Shishara College Government, Salem. Coming to plasmid, that is, plasmids or vectors. So what is a vector? Vector is a DNA molecule that is used to carry a specific DNA segment into the host cell. So the DNA which we need to transfer from one cell to another cell is transferring by a vehicle, that is carrier, which is known as vector. So the important vector available is a plasmid. Plasmid is a small circular double-stranded DNA that is distinct from other cells. Features of vectors. Vectors act as a vehicle to transfer genetic material from one cell to another for different purposes like multiplying, expressing, or isolation. Vectors are used as a tool in molecular cloning procedure. So it is inserting desired DNA into the host cell. Since I am introducing desired DNA into the host cell. The process of gene transfer also differs from one vector to another vector. Some of the vectors directly enter into the host cell and incorporated into the host DNA. But since some vectors just pass the genetic material into the host cell and they recover themselves. It is not completely entering into the host cell, only the genetic material is entering. Even though vectors are usually DNA sequences, viruses and other particles also function as a vector. Vectors can be reused for multiple processes. Since it is recurring, we have already seen. A cloning vector is a category of vectors that are essential for cloning procedures. These vectors have different sequences that enable them to initiate replication in host cell as well as propagating with the host. This plasmid vector is a small extrachromosomal circular DNA molecule. It is capable of replicating autonomously within the host cell. So no procedures involved, it can be able to replicate themselves. There are also many vectors involved in the recombinant DNA technology, and plasmids are the most important thing. Plasmids are widely used vectors in all the three domains of life, and they are frequently used in bacteria and yeast. The autonomous replication of plasmid is made possible by the presence of genes and sequences that can initiate plasmid replication independent of the host replication cycle. Bacterial plasmid contain a ORI sequence that not only control plasmid replication but also determine the possibility of coexisting with the same host cell. So it should live along with the host cell. So for this particular coexisting factor, this ORI sequence is helpful. This is naturally occurring plasmid and can be modified by in vitro technology also. The scientist Cohen in 1973 reported this plasmid, and it is not that much essential for the growth of cell, but it can determine the replication factors. They have a special origin of replication. They can pass from daughter cells even during cell division. They carry a marker genes, and they are of many types: relaxed plasmids, stringed plasmids, conjugative plasmids, and non-conjugative plasmids. This can be isolated in a bacteria also. Coming to PBR 322, PBR 322 is the full form: P for plasmid. They are Bolivar and Rodriguez. They are the scientists who discovered, and 322 is a numerical designation. It was constructed in 1977 in the lab of Herbert Boyer at the University of California in San Francisco. This is synthesized plasmid and the vector first artificial plasmid to be constructed and used. For most of the studies, PBR 322 is the major role, playing a major role. It is containing 4,364 base pairs. It is of completely sequenced, which means the whole sequence of PBR 322 is known and already studied. It is having molecular weight of 2.83 into 10 to the power of 6 Dal. It is commonly used plasmid cloning vector that is used in prokaryotes. PBR322 was designed to overcome the limitations with PBR322 with PBR3 12 and 13. So previously we have derived PBR 312 and PBR 313. The vectors have certain discomfort and scientists discovered in order to overcome these limitations, the V22 is discovered after that. Both of which having extraneous DNA sequence and restriction enzyme cleavage sites that affected their functions as vectors. The structure of PBR322 was designed to maximize the number of restriction enzyme cleavage site on the vector and to minimize its size. So the size is minimal. The vector contains 21 unique restriction enzyme cleavage sites, 11 of which are present in TCR and AP genes. This is the structure of PBR222. It is having special marker genes, amphicycline and tetracycline, and every plasmid is having origin of replication and other restriction sites. The main characters of PBR322 are it is commonly used plasmid. It contains the genes of resistance of amphicycline and tetracycline and can be amplified with the chloroalkyl. Restriction sites like BAMHY, HIND3, SAL1, PVU1, PVU2, PST1, ECO, R1, CLU1 are extra restriction sites present in this particular plasmids. Selectable markers like antibiotic resistant genes or of amphicycline AMP are and tetracycline TETR are present. Different plasmids have different selective markers, but most common includes these antibiotic resistant genes and produce of B-galactosidase enzyme, beta-galactosidase enzyme. So it is also having origin of replication, that is ORI, and it codes for the proteins which are involved in the process of replication of plasmids. So coming to the selectable markers which is specifically present in the PBR322, we have discussed amphicycline resistant gene site and tetracycline resistant site. The amphicycline gene codes for beta lactamase, which can be used for screening microorganisms when a foreign DNA is inserted into a plasmid. So, when a foreign DNA is inserted into a plasmid, we need to detect the microorganism. That process is possible when there is an amphicycline resistant site. This particular tetracycline resistant site, this gene degrades the antibiotic tetracycline and can be used for screening microorganisms. These antibiotic resistant genes are useful in screening of organisms after the cloning process. The functions of amphicycline resistant gene, it confers resistance to amphicycline, allowing for selection of bacteria, and the mechanism involved is it encodes with the beta lactamase enzyme and break down the amphicycline, rendering it to be ineffective. Similarly, tetracycline resistant gene confers with the tetracycline, allowing the selection of bacteria that have been taken up in the plasmid. This particular gene encodes a protein that binds with tetracycline molecule and preventing its inhibition in the protein synthesis. The purpose of selectable marker is selection allows the selection of bacteria that have been taken up in a plasmid, successfully identifying the bacteria that is successfully transferred with the plasmid, elimination of bacteria that is not taken up by the plasmid. So the pre-process selection, identification and elimination is done by these markers. Different antibiotic resistant genes act as a restriction site and to ligate foreign DNA and for selection of transformers. The gene where the foreign DNA is inserted becomes inactive. We have alternate selective markers also. Mostly they have the ability to produce some color after reacting with the chromogenic substance. The alternative markers are used in the case of different recombinant from non-recombinant. Example, gene coding for galactosidase. When a foreign gene is inserted between a gene coding for beta-galactosidase, the recombinant cell does not produce enzyme beta-galactosidase due to inactivation of the gene. In the presence of chromogenic substrate, the non-recombinants form a blue color colony and the recombinant form the colorless colony. Restriction enzyme site. Around 40 restriction enzyme sites are present in the genome of PBR322. Almost 11 different restriction sites are present in the region of tetracycline resistant region, and 9 restriction sites are present in amphicycline resistant gene. Screening of recombinant containing PBR322. Consider that the restriction enzyme BAMHI is used to cut the plasmid at the specific portion. It lies in the tetracycline resistant region of the plasmid. Now the recombinant molecule of PBR322, which includes the newly inserted DNA molecule, will be sensitive to tetracycline. But this recombinant molecule is resistant to amphicycline as the amphicycline resistant gene is fully functional. Hence, when the recombinant cells are plated on the medium containing amphicycline and incubated, the colonies that appear on plates containing a medium with amphicycline are transferred to the colonies containing the cells with a newly inserted DNA molecule. The replica plate technique is used to confirm the transferred colonies. To confirm that the colonies of amphicycline containing media are transferred colonies, they are placed on the plate containing medium inclusive of tetracycline. The colonies that do not grow on the medium inclusive of tetracycline are transferred colonies, as the transferred colonies are sensitive to tetracycline. So what is the uses of this particular PBR322? It is helping in gene cloning, and it is helping in protein expression and helping in the DNA sequencing. Cloning processes. Cloning processes involve digestion, ligation, transformation, and selection. So PBR322 plasmid is digested within the restriction enzyme such as EcoRI to create a linear fragment with sticky ends. The target DNA fragment is ligated into the linearized PBR322 plasmid using DNA ligase enzyme. The recombinant PBR322 plasmid is transferred into E. coli bacteria. The bacteria that have to taken up the plasmid are selected for using amphicycline and tetracycline. Expression. Expression involves transcription and translation. The targeted gene is transcribed into mRNA by E. coli RNA polymerase, and mRNA is again translated into protein by E. coli chromosomes. Advantages of PBR322: it is having high copy number, which allows the high level of gene expression. So more copy number, more gene expression. Easy to manipulate, relatively small plasmid we can easily manipulate. Well characterized plasmid, it is popular choice for molecular application. It is having certain limitations. Limited insert size. PBR322 has limited insert size which may clone large area. Instability, sometimes it is not having stability, and plasmid loss or rearrangements are present. So we have discussed about PBR322, a plasmid which is having a major role in gene cloning technology, and hope you all are clear about the PBR322. Thank you.